My name is Liz Schickling, and I'm a marketing manager here at Worldwide Clinical Trials. Before we kick things off, I wanted to go over a few things. All attendees are on mute, but please use the question windows to submit questions at any time pertaining to the presentation, as we will be reserving time for a Q&A session at the end. This webinar is being recorded, and all attendees will receive an email with a link to access the on-demand recording. I would now like to introduce the 5 experts who we will be hearing from today, who represent many different areas of our organization. Dr. Michael Murphy, the Chief Medical and Scientific Officer. Allison House, our Director of Business Development. Steve Unger, Senior Vice President and Laboratory Director of Bioanalytical Sciences. Leslie Weatherall, our Executive Director of Rare Disease Franchise, and Sherilyn Adcock, Executive VP of Medical and Scientific Affairs, Early Development. Before we begin, before we drop in on the experts, we'd like to take a quick poll of the audience. Dr. Murphy, would you like to elaborate on today's poll question?
Fantastic. Thank you, Liz. Welcome, everyone. Thank you for joining us today. Before we begin and intrude on a discussion which is already ongoing, we wanted to calibrate your interest a bit. And so the quick poll question is designed to inform us as to your perspective and business strategy. So as you look at your organization and the R&D initiatives which are ongoing, do you find early phase work more or less critical to your strategic program design? And by that I mean, by early phase I mean first in human target engagement and proof of concept investigations. By more critical, we mean you base most, if not all, of your strategic program decisions on the information that you extract from the first few clinical studies. Or by less critical, we mean although you acknowledge early phase work to be highly informative, you also see inherent limitations in earlier phase clinical trials, and therefore you create flexible, adaptive R&D strategies with many branch points. That is, no one study would be definitive. So as you think about it from your perspective, how would you score it? More or less critical? Liz, if you could, please. Okay. Fantastic. Well, a lot of interest in this topic, of course, and as there should be. Let's drop in on the experts and see what's going on here. So, Allie, I know you're online. I want to ask you a couple of questions. Now, Allie, you come from a pedigree of both clinical operations, which is quite extensive, as well as business development. So let me ask you, so given the changes we've seen in R&D efforts the last few years, are you encountering more opportunities for strategic CRO services for earlier phase investigation, or is the field still pretty much dominated by transactional service requests? So I guess I'm saying, Allie, are you seeing more strategic partnerships? Or are you seeing where people are developing compounds to inflection points, you know, or not? How do you see it? Allie, if you could, please. Okay. Well, while we're waiting for Allie to come on again, you know, related to this is whether or not partnership conversations with clients are more or less frequent. Sherilyn, I know that you have quite a bit of activity on both the early phase development as well as the BD initiatives. You know, what's been your experience today?
Dr. Murphy, this is Allie. I'm so sorry. I was struggling with the connection here. I do apologize. I don't want to step on Sherilyn's toes. I know you posed a question to her, but I am happy to proceed.
Yeah. Go right ahead, Allie. I think we were all on mute by the organizer, but I think we've got it.
Okay, great.
Well,
Okay, so just to, with this question that Dr. Murphy has posed, I think this is an exceptional question. And based upon this, I think the type of service that we see coming in from potential clients is really, it's based largely upon the demographic of that sponsor company. In many of these situations, we find that the small biotech organizations are looking for more of a strategic partnership in nature. So this is inclusive not only of the implementation and conduct of the trial or program, but also requires extensive consult and navigation of the clinical development process. So we may start as early as support in assembly of the initial IND submission documents, all the way through the proof of concept studies. Contrary to this, though, we do often receive requests from those companies that are larger, well-funded pharma companies, and they tend to lean more towards a transactional nature for study conduct.
So Sherilyn, you come from the same background as Allie, strong operational experience, but also business development responsibility for early phase services. Are you seeing the same thing?
Yes, very much so. I do see the studies becoming a bit more complex in phase one, trying to capture a lot more data in single studies. And we can talk a little bit more about that later, but I think Allie is right on, hit the nail on the head. It seems like the smaller companies, the biotech, and some of the mid-sized companies, which we're a nice fit for, because we have kind of a seamless operation between phase one and into the phase two and into some of the patient studies. You'll see those individuals and those clients that are looking to carry their project forward, not license it out after phase one, but carry it forward. You can see the excitement build as they begin in phase one, start getting a lot of that information and capturing a lot of that data, and then start moving forward and seeing that forward progress into the early patient studies in phase two.
Well, Shirley said a neat thing there, which we're all aware of. So, most CROs are tending to organize themselves into earlier versus later phase activities, which of course is understandable. But is there a need for a different type of business model that's more creative to exploit what you've just said, the changing role of R&D and the more strategic relationships? How do you see that?
Well, I know with a number of our companies over the last, I would say 3 to 5 years, we have seen the involvement of medical and scientific affairs. So it's clearly not just a transactional, here's a protocol, let's move forward with it. We've seen companies come in that may be in the later stages of phase one, or they may be in the very early stages looking for that medical and scientific affairs expertise to come in and help with things such as with drug-drug interactions with CNS drugs, titrating drugs up to a steady state so that you can do your interaction aspect of the study, and then titrating down because some of these drugs are a little bit more dangerous to deal with and a little bit more toxic at times. So we have to be very careful at how we proceed, particularly in normal healthy volunteers with some of these compounds. So I think the companies are needing that expertise out of medical and scientific affairs and looking forward into that phase two, which we need the later phase experts to help us with.
Leslie, what about the operational services? I know you're heavily involved in both early and late phase activities. Are you seeing the same type of development that Sherilyn and Allie have alluded to?
Right, yeah, I would add that, we've worked on sometimes similar indications multiple times over. We see very similar protocols over and over again. And we can help avoid some of those pitfalls that we see in the earlier trials when we get into the later phases. So we have the ability to pull in experts, across the different facets of our company through labs and early phase through Sherilyn. And so we can work together to help transcend the type of silos sometimes we see in organizations, where we can really work together and help each other plan for the next phase of study.
Go ahead, Cheryl. Please.
I think one of the things that is important to consider, especially as you add more testing and potentially using outside vendors such as cardiac core labs, EEGs, specialty lab facilities, and even patient populations in these early phase studies. You have to consider your vendor and how qualified that vendor is, and that's where quality assurance in your company plays a very key role in helping to qualify those vendors to ensure that your client is getting the best service and the service that the FDA is going to see as a valuable and a qualified service to provide, to give them the data they need.
You know, I'm hearing a theme in the undercurrent here. It talks about complexity. So how many is too many questions for a first-in-human study? So how do you see that, Sherilyn?
Oh gosh, I can tell you, having spent decades in this particular area, particularly in the phase 1 area, I have seen over the past 5 to 7 years these studies in phase 1 really evolve and becoming more and more complex, asking many, many more questions in a single protocol. For instance, 15 years ago, you would have a single ascending dose study. You would evaluate the data from that. You would go into a multiple dose study, and you evaluate the data from that. Then you would consider patient populations and special populations, a cardiac, intensive cardiac evaluation. Now we're seeing all of those kind of folded into one protocol, and that's certainly more common to see: single ascending dose, multiple ascending dose, a food effect, a special population, either a patient population or elderly subject cohort, intensive cardiac evaluation, and even EEG added all into the same protocol. I think the biostatistical applications certainly these days can support that. I would say 15, 20 years ago, we probably caused a lot of angst in the biostatistical divisions when we tried to do too much in one protocol. The challenge becomes in the logistics, and that's where you want your operational group. And we have a feasibility group that meets, and we really try to work out the timelines of what's going to happen, how this is going to flow through, when the data needs to be to the client so that they can make the decision whether to move forward in the next dose, so our PIs can look at that data and ensure that safety is, the utmost importance.
Now, Leslie, I'm hearing yet again an operational theme in the background. Is it possible to design a study that no one can execute? Or how do you see that one?
Yeah, so sometimes what we see in early phase trials that are in sick patients, such as a phase 1 oncology study, is that we have really complicated assessments, PK draws, overnight stays, and this is much harder in these sick patients, than it would be in a healthy volunteer. So we really have to think about the patient's experience, right? Also in rare diseases, sometimes it can be really difficult for those patients to participate in all of the requirements. So the burden sometimes can be on the patient, and we just really need to think through, we make it so difficult for the patient to comply with a protocol. Do we really need to rethink the schedule of assessments, and how could we make it a little easier, perhaps use home health care or some other mechanism to allow different ways to retrieve the data without forcing the patient to come back and forth to the clinic.
Well, given that this good discussion, that there's an expression out there that the physicality, if I may use this word, of phase 1 units have moved away from the prototypical dormitory setting that all of us remember from 30 years ago to something that sounds more like an intensive care unit. So, Sherilyn, are you seeing changes in staffing patterns? Are you seeing needs for different types of services? I was fascinated a few years ago. I had never encountered this concept I call a drug in a bottle, where we could avoid making different unit strengths early on and just have a much more flexible, dynamic approach to dose ranging. So My question is, are you seeing a change in the type of service and the type of people you need to hire into these early phase units?
Um, in many cases, yes. You still need the individuals that can look through this from an operational standpoint and make sure you don't have an ECG on top of PK draw on top of vital signs and such. But there are some special services where we either need to bring in consultants, for instance, if for CSF collection, and a study where we're comparing maybe pharmacokinetic blood samples to cerebral spinal fluid samples to see how the drug's crossing the blood-brain barrier. Clearly, we need anesthesiologists. We need special equipment that we bring in and individuals that can manage that equipment to capture the all the assessments that we need in something like that. One of the things that we're seeing very much in the last probably 3 to 5 years is we've seen the pharmacy services with expand. There was a guidance put out by the FDA with respect to phase 1 cGMP compounding of phase 1 investigational drugs. This has opened up quite a large area, and the big advantage of this is that in phase 1, where as we discussed earlier in our conversation, many of these drugs are killed at the phase 1 level and do not move forward. You can save usually 3 to 4 months in time, and you can have these smaller batches made of product, and they're made on site in a cGMP environment that follows this phase 1 guidance. At this time, we have approximately 9 studies going where we are providing that early phase compounding service or manufacturing service. That includes powder and bottle for reconstitution at a later date. That reconstitution may be with another manufactured product to mask the taste. We have capsule filling down to 3 milligrams per capsule, dry powder inhaler capsule filling, suspensions that are made just prior so you don't have to worry about long-term stability. So a number of the projects are utilizing that service to help them move forward in their timeline a little bit faster. It'll save them some money long-term, particularly if you don't have to worry about the large 10,000 tablet quantities you might need for a study from a contract manufacturer at that time. Now, contract manufacturers certainly have their value and their place in phase 1, but for some of the very early first-in-man studies, we're finding this is a path of the cGMP manufacturing. This is a path a lot of companies are taking.
You know, Sherilyn, I didn't realize how important this was until a few years ago. You know, many of the clients we have the pleasure to work with, they're small companies, and they really don't have the investment capability to, you know, full kind of CMC-related portfolio for first-in-human before they know they have a product that has value. So I really think the pharmacy service particularly is something that's so nice to have in early phase clinical research. Very nice indeed. Now, you know, related to this, everyone, you know, I always hear is, you know, what is a fit-for-purpose assay? I didn't know about this idea until a few years ago. And Steve, I mean, why would a company ever use it?
Thank you, Mike. Most of the work that we do with fully validated methods. The lab's been around a long time. We have over 2,200 of these validated methods. They're good for all kinds of work, co-med, DDI, any type of clinical pharmacology study that clients are interested in. Sherilyn mentioned we have CNS biomarker assays for looking at drug effect and penetration into the CSF. What we do in the fit for purpose is really, it's an assay that doesn't meet the full regulatory rigor as defined by the FDA and EMA guidance. It's done under a qualification plan. We have an SOP for it. It uses written predefined figures of merit. We agree with the client what that needs to be. Sometimes these are referred to as tiered assays, qualified assays, non-GLP assays. Um, it's less rigorous than a fully validated method, but it's something that allows you to answer questions that you might not be able to answer very well or very cost effectively with fully validated methods. So sometimes used to talk about off-the-shelf immunoassays or metabolomics kits. Basically, LC-MS can measure thousands of components simultaneously. So the question is, what actually needs to translate in terms of biomarkers into something that's important to measure? And the same applies for metabolism. Metabolism-mediated toxicity, MIST ICH guidance is really required you to look for unique or disproportionate metabolites at steady state exposure. So that means going across human back to animal to make sure you have safety coverage in at least one primary toxicology species. And as you can imagine, if you have a drug that's extensively metabolized, you've got a lot of work to do. So, it really allows you to kind of go after these directions and answer these questions in a way that's efficient, cost-effective, and can be done earlier so that you're measuring the right metabolites and not the wrong ones. You really want to be measuring parent and active metabolites for your standard fully validated clinical assay, but you do need to get this other information to make sure that you have the safety coverage that meets MIST criteria. Sometimes doing this really makes it difficult because some of these are biologically expressed, so you can't have a certificate of analysis, which in its own sense precludes you from being developing a fully validated method. There are a lot of other potential uses of it. We sometimes see clients using it in the SAD study to estimate the renal clearance urine to get a good handle on what the assay should be in a fully validated assay for later, say, an MAD study. Free fraction assessments done early for protein binding could be done this way. You don't want to do it kind of in an ex vivo protein binding study. Penetration questions basically can be covered under this type of assay. So anything that's kind of not a primary objective within a clinical study, but an exposure measurement that needs to be made to provide useful information to guide a development program.
So exciting, Steve, and so strategic. When you first mentioned this to me a few years ago, I thought you were just speaking colloquially. I didn't realize there was such a literature on the topic, and I really am excited by that capability. But let me ask you, have you had an experience in which the fit-for-purpose approach was off the mark? It was misleading. Does that ever happen?
Yeah, I think this tends to happen more if you're just buying a commercial off-the-shelf kit assay and then trying to translate, buffer calibrators to a matrix method. With the exception of the Biocrates kits that we've used, and this is a kind of an omics translation kit, we tend to build our own assays and stay away from the kits. That way we really know the product that we're implementing, and obviously then we can make corrections. We're always pretty big on adding stable label to what we build here, and so if there's quality that needs to be added to the method, we're not going to go cheap on this. We're going to make the investment and do it. So as I mentioned before, we do have this SOP on how we manage fit for purpose. We have a form that clients work with our head of method development to fill out. It gets reviewed in a qualification plan. So it's important to do this, have a priori criteria. You can't kind of judge this after the fact. So if you set those figures of merit in place and then execute against those. Probably the most common complaint that we actually get are when people go this way, they find important data and then they wish that it had been fully validated and they repeat it later. Yeah.
Unbelievable. Well, lastly, let me ask you something. So, does your operational strategy, change? Do you do things differently when we have what I would call a fit-for-purpose stratagem in place, particularly for some of the orphan indications that you're so heavily involved with?
Right, yeah, so sometimes when we're going in rare disease populations, we're, skipping some of the steps, kind of going straight into phase 3 studies, and so we end up with a lot of assessments for those patients. Like I said, that can be very difficult for them to, be at the clinic as long as they need to be, or they may have some kind of physical limitation that doesn't allow them to be on site for long periods of time. So if we can use home health care for these kinds of samples, PK analysis, any kind of other, lab samples that we need to collect, it's really helpful to the patient to be able to comply with the protocol and make sure that we're collecting all the data that we need to collect.
I tell you, Steve, some of those micro-sampling techniques that you folks have developed are so useful to us with the kiddies, really exciting technology. So we now have this question, Leslie, so since phase seems to matter for strategic operations, is it true or not that the type of monitors that we need for early phase differ from those that are in late phase? Kind of like fewer but more trained with a deeper background? How do you see that?
Right, yes, especially in early phase studies, I think we need to ensure that the monitor, can be on site as much as possible for that first patient at that site to help the site, understand the protocol, if the drug has to be reconstituted or anything like that, really helping them to be a support. So that's something that we're seeing as more of a trend that, we try to encourage our sponsors to utilize our monitors in that way. And then if we have a CRA that has a nursing background or a study coordinator background in that phase 1, phase 2 type study, it's really helpful for them to be that kind of support to the site. When we get into the later phase trials, like the phase 3s and 4s, the endpoints are streamlined and the protocols refined, so we don't see that we need, that kind of support or that kind of experience so much. But like I was talking about before, with rare disease studies where we're stepping into those later phases a little faster, the sites might not have as much experience with clinical trial, like good clinical practice, and so then we need to be able to train and support the site, help them understand the requirements, regulatory requirements, etc., to help them be successful and make sure that the quality at that site is high. So a lot of times they're only having maybe 1 or 2 patients at a site because of the low incidence of a disease. So we just really need to make sure that they have adequate support and training, and that the overall, quality of the study is there.
Well, thinking about the outreach to the PIs, which of course is key. So when do you need to be involved in protocol design, Leslie? I mean, sometimes I feel we bring you folks in too late into the discussion. How do you see it?
Yeah, so oftentimes, when we're talking with clients, they've already reached out to a few KOLs or lead PIs to discuss the protocol, and that's pretty typical. But what we find in rare disease protocol design is that it's becoming increasingly important to seek the feedback of patient advocacy groups. Then we're able to really get the valuable feedback from the patient on what is going to be workable in their life. That's not just trying to figure out how we're going to assess the disease, but how does the disease manifest itself in their everyday life, and what limitations do they have, and how can we work around that. So gaining the feedback from that patient advocacy group, you're really getting the direct feedback from the patient on how that schedule of assessments might or might not work, and then we can tailor our protocol to make sure that they can comply and we don't have any early dropouts as a result of maybe too complicated of assessments.
So I hear you, very early and very involved, even in the initial concepts.
Right, yeah.
Yeah. Now here's a topic, team, that I know all of us have a strong opinion one way or the other on. So Allie, is it patients or is it normal volunteers for a first-in-human study? What are you hearing? What are you hearing in that regard?
I think that's a great question, Dr. Murphy. At this point, it's really important that we work with our clients to pay attention to, what we'll optimally focus upon in the early phase studies. Occasionally we look at this, we may see that there's a development option that may be differently based upon the experience from, the responsibility for product development, our own individual histories, or as well as, as a CRO as a whole. most CROs will report similar sentiments. It's not really unique to WCT, but it certainly is a reflection that all professionals, within the pharma industry arrive at these positions with common lineages. I think, when we're looking at with these clients, reviewing these protocols, Worldwide takes pride in taking a full review from all facets. We look at it from a commercial standpoint, a clinical standpoint, a medical and safety standpoint, and an operational standpoint to determine what is the best study design to get more bang for your buck here, basically, and ascertain the information that's needed. So in looking at this, do we go straight into patients or we stay with healthy volunteers? It's really contingent upon the type of data that we have that's preceding this, whether it be a first-in-human study or if we're looking just beyond this, and determining how to most optimally design the cohorts to support this.
Well, I have such a strong opinion on it, but Sherilyn, how do you see it? Normals, patients, what's the advantages and disadvantages?
Well, certainly there are definitely disadvantages to going directly into patients, and I think Leslie has pointed some of those out at this point. The access to those patients, the timing of getting the information that you need, and obviously you want the information as fast as you can get it. If the drug is safe, if we can put it in normal healthy volunteers, I think there's a lot of value in starting with normal healthy volunteers, looking at maximum tolerated doses and such in normal healthy volunteers. However, at some point you will find that with, as you know, Dr. Murphy, with some of the CNS drugs, what is a maximum tolerated dose in healthy volunteers may be a higher dose or it may be a lower dose in patient populations. So clearly something that you have to look at, that translational aspect of what do you do after you get through with your healthy volunteers. And that's the one thing we're seeing in a lot of these studies now is the single ascending dose is in healthy volunteers. Multiple ascending dose starts in normal healthy volunteers, but then transitions into cohorts of either special populations or patient populations.
Well, Leslie, I know that parents, for example, for the developmentally disabled child, how do they see this? Are they open and amenable to kind of a first-in-human study with their children?
Right, yeah, so oftentimes rare diseases are diagnosed in that pediatric population, so we're seeing more and more rare disease studies going into the adolescent and then younger population, sometimes as different cohorts. So there is some resistance for parents to enroll their young children into clinical trials, but they might not have any other option. So the more information we can give to that parent or caregiver about the early safety data, and so that they can make an informed decision on whether or not to put their child in the trial, is really helpful. Sometimes we see that as kind of a handout about the study, similar to information you put in an informed consent, just to let them go home and think about it and come back and make a decision. I think it's really helpful. The more information we have like that to give to the parent can help with the enrollment. Otherwise, we could be stuck trying to find, patients to enroll if we don't have enough information to kind of put their mind at ease.
And Steve, I know that not everything is always first in human, and particularly we have under PREA, pediatric study plans do require that we expose children to products in adolescence. So what are the unique contributions that you guys make in bioanalysis in that regard?
Yes, Mike, you'd mentioned the microsampling, and we're doing a study right now. We've looked at every technology that's out there, from dried blood spots to mitro to capillaries, plasma and blood, wet or dry, and we can basically scale any method that we have. Electrospray is a concentration-dependent detector, and so if you don't dilute the specimen and keep it in its original form, it actually helps significantly in sensitivity requirements. So we can take and adapt and still hit the same sensitivity, detection sensitivity, by scaling the volume down. And so that's one way that we contribute to these types of studies. And as I say, we've done even studies like this in addicts where the vascularization is compromised, and we're doing finger pricks in a regular adult, pediatric rodent studies, things like that, where the microsample is a required part of the analysis. The other thing
I hadn't thought about that. You know, in patients who have compromised vasculature, I hadn't thought of that. Very interesting. Yeah.
The other part about rare populations is in the last year and a half, we've started doing a GENA-based, formerly known as sequenome, pharmacogenomics for helping to call out our population in San Antonio, particularly for SIPs and transporters. So, you know, hopefully that will add some value. If clients that want to do studies and say 2D6 poor metabolizers or 2C19 ultra rapid metabolizers, that we can screen that population and have them available for enrollment.
Such an important service. You know, many of our clients are doing adaptive trial designs, adaptations on dose, adaptations on genotypic or phenotypic features. So having a strong bioanalytic capability, such as you've implied, is really neat in terms of strategic program development. Very exciting capability. And the integration between early and late phase really is an important discussion. So as we sit back and look at it, everyone, where do we see early phase clinical research activity heading? Now, if we thought about it a bit, what it seems to me is that there's a lot of advanced technology out there, people targeting very refined patient phenotypes. So it's a very changing landscape. Many of our clients are very interested in establishing what I call value very early to a critical inflection point because they want to make some key program decisions. So an ability to integrate all the services we've talked about today in an innovative program rather than a protocol seems to have significant merit. As we heard from Allie, and Allie, you know, you made a nice point in this. This interest obviously varies by sponsor, and we all know it also is different by therapeutic area. You know, contingent on the area, the target, the sponsor, this sentiment can be you know very different. Also, as Sherilyn has pointed out beautifully, more questions, more assessments, more procedures, and specialized services. I really get back to this pharmacy service, and also we didn't talk about it, Sherilyn. I think the dietitians you folks have on board are very helpful. You know, food effect studies are critically important to inform later phase programs. And you can hear, you know, the way we all started in the business in dormitories is now rapidly changing. It really is something that's more closely aligned with an ICU. And Steve, I mean, just listening to you, the techniques, the way of sampling, you give us such flexibility in terms of the types of experimental designs we can consider. The ability to integrate PK and PD, you know, it's all target engagement. I mean, those are the questions we're getting repetitively. You know, this compound, get in, hang around long enough, have a handshake that looks interesting, and then I'll take the next step and look at efficacy. And the ability to move quickly, efficiently, get information very early to move to a later stage, you know, seems to be key. And then finally, Leslie, you got to bring all this home, you see. That there is a challenge here about integrating all of this activity into a later phase setting in a seamless fashion, a totally transparent and seamless fashion. And it reminded me a lot of kind of a matrix organization that many of us worked in pharma. You know, when you had a product under evaluation, you would be on a team from the IND enabling, sometimes even before that, but certainly by the time of IND enabling studies. Whether you were in the commercial space or the development space, you'd be on that team. And that organization would stay together for the entire life cycle of the product. And this seems to me that type of process, that's the transformative part of R&D in terms of how we're doing things. And so, really, I think the differentiation in R&D today is not only in the service or the professionalism of the staff, which is really exceptional, but it's how that process is being facilitated by academia, by pharma, and by CROs, frankly. So, fascinating discussion. We now have some opportunity here for some questions from the audience. When we've done these webinars, there's usually quite a bit of activity on the Q&A side. So what Liz and I want to do is encourage everyone to give us a question. We'll see if we can answer it, and we'll take it from there. In fact, we've got our first C question in here. And so here it is. So do we think that the current CRO model is going to remain viable 5 years from now? Are we basically set up to engage the R&D space with highly differentiated services where we integrate early and late phase activity, systems, processes, and so forth? So Leslie, Allie, team, how do you see that one?
Yes, I can take that one. It's Leslie. So one of the things that I'm seeing in the rare disease development space is that we see more and more studies in rare disease, and obviously Dr. Murphy and I are spending a lot of time talking to clients about that. It's great news for everyone that's affected by rare disease because it has the promise that this is an expanding area of research. But we need to really tailor that strategy toward the long-term commitment of those patients. So this is something that we're talking about with our clients, that we need to take them from the phase 1 all the way through to post-marketing, and sometimes longitudinal or natural history studies, because perhaps there hasn't been any other research conducted yet, and so we really need to gather as much information as we can and help our clients with going through the steps to do so. Our commitment to those patient populations is long-term, so we have to start with that in mind when we're planning out the full development strategy.
And Allie and Sherilyn, I know both of you have had these ideas that maybe we're organized in a less than optimal way. Maybe we should be thinking more of an integrated early phase versus late phase footprint. Allie, you had some incredibly interesting ideas working with the team in San Antonio in that regard. How do you see that?
Well, Dr. Murphy, it's a very good question that you ask. I think within this, what we have done as of late is when we see opportunities coming in that have the ability to move, or we're looking at that strategic relationship in which we're moving from our early phase to our late phase divisions, we typically assign a therapeutic and operational advisor to support the conduct of the study from the time the RFP is received, working collaboratively with our phase 1 groups from a late phase perspective to ensure that we have the same types of resources that can kind of follow this or navigate through the phases of the studies. In addition, we look at, for studies in which we're going outside of the healthy volunteer base, we engage, when we're looking into patients, we certainly engage investigators as well and site staff that can also work with us to move through the early phases to the late phases. So we've had a couple of joint opportunities in the past couple of months that, are going quite well utilizing this model, and I think we'll probably see quite a few more coming in. So Sherilyn, anything to add from your end?
Well, I think the communication is very important in bringing in these advisors that will follow this through late phase, getting them involved early on, even from the first cohort. Yesterday I sent out an email, giving an update on when the first in man for a new Alzheimer's disease product is coming out. We're doing the mock preparation today at the pharmacy, so it's a CGMP for phase 1 product, but we're keeping the individuals that are involved in this from a late phase scientific design standpoint, involved in the whole process. So they'll know what AEs are occurring, and this process moves very fast, as you know, in phase one. We get our samples over to Steve at the lab within a matter of hours after the samples are frozen, processed, frozen, and drawn. Steve's group can handle the bioanalytical assays, so within a few days, we can have results for each cohort and each dose escalation and know the labs that are involved, the diagnostic labs, the bioanalytical results, the PK results, and we can move this forward. And I think it's very important for the late phase clinical lead to be involved in that and to understand all that communication and really have the story from chapter one on this drug.
You know what's really exciting, Charlene, and I've been on the tour with you a few times, and it always amazes me after all the years we've worked together. Every time we're together, I get another idea, another notion, a new thing you should consider for experimental design. So just at that level alone, it's clear that 2 and 2 equaling 6 when we get together on some of these programs, and very exciting. And Steve, let me ask you another question that just came in here. So it has to do with whether or not pharma has experimental medicine units and translational medicine units, and so how do you interact with them? What's that experience been like for you?
Yeah, I guess when I've worked in pharma companies, we did have well-established translational medicine groups. So, you'd look at developing more discovery-based biomarker and then translating it into the clinic, and I think this is really going on a lot in companies like Metabolon or companies that are selling metabolomic services as an LCMS-based, say, alternative to an LBA assay. But just the idea that there's so many potential biomarkers out there, and that's actually how this fit-for-purpose term originally came about, was to handle this demand for translational biomarkers in translational medicine. So as Carolyn mentioned, if we don't have it, we partner with experts that do have it to help people do translational medicine and help to get to proof of concept, because a lot of it's moved away from just DMPK exposure and safety, the typical phase 1, into efficacy, and people are interested in knowing if they have target engagement, and they have a mechanism of action that they want to test in the clinic, and they want to find that out as early as possible. So I think that's one of the changes that we're seeing in the industry that laboratory CROs need to adapt to and make sure that they can cover. We can cover about anything in HPLC, fluorescent LCMS based, including assays we've done up through the potential to do any signature universal peptide or oligos. So we have ways to cover from small to large molecule. We are predominantly an LCMS-based lab.
Well, interesting comment there, Steve. We've all seen that. The technology now is so advanced. It isn't simply small molecules, as you've indicated. So incredibly and excitingly different world we've lived in compared to the last 10 years. Well, everyone, we're running near the close of the hour. I think we've covered a number of interesting topics here today. I think it heralds the future of R&D for innovative discovery, development, integration. And on behalf of everyone at WCT, and really want to thank you for letting us join you today for this good discussion. And if you could, Liz, take it from here.
Thank you, Dr. Murphy, and thank you to our experts for letting us drop in on you today. This concludes our webinar. If you asked a question that we were not able to get to, we will reach out to you individually. As I mentioned earlier, this webinar has been recorded, and all attendees will receive a link to the on-demand. For more information about Worldwide, we invite you to visit worldwide.com. We hope you enjoyed today's webinar and have a wonderful rest of the day.
Great, thank you.